He is a PhD candidate at CU Boulder in the Department of Ecology and Evolutionary Biology, and he studies evolution and development in lamprey, sharks, and other fish. Zach currently has a BS and BA degree from the University of Idaho, where he graduated with a double major in biology and Spanish in 2016. Outside of the lab, Zach is a huge nerd for cooking, cheese making, linguistics, and video games. And also joining us tonight is our navigator, Fisk's very own John, a recent graduate of CU with a BA in astronomy. Tonight's talk falls under our Science of Sci-Fi series and is titled The Lego Box of Life, Genetic Engineering and the Science Behind Jurassic Park and Gattaca. Definitely check out our other talks on this YouTube channel under all of our categories, the Science of Sci-Fi, Science and Society, and Climate Change in Our Backyard. So with that, Zach, please take it away.
Thank you very much, Amy. And a big thanks to all of you that were able to attend tonight. I'd also like to thank Fisk Science Under the Dome and the University of Colorado for being able to give this talk to you guys and talk about some really cool science for that matter. I think it's important to acknowledge as well that they're long before these institutions ever existed. And for that, I'd like to recognize these lands as part of the Ute, the Arapaho, and in more recent history, the Cheyenne. With that said, let's get started. So yeah, tonight we're going to talk about genetic engineering, what we know about it right now, what tools we use for it, and we're going to compare it against some science fiction movies, see what they get right and not so right. So for that, we'll be looking at the 1993 movie Jurassic Park and the 1997 movie Gattaca. So for starters, I love science fiction. I think it's a great way for us to explore concepts and ideas about technology in the future. It lets us retroactively think about what we thought about a technology based on the way we imagine it playing out in the future. So before we can talk about genetics and science fiction, we need to talk about our historical understanding of genetics, what we know about it nowadays, and combine those together to get a sense of what we think it could become. So even before we knew what DNA and genetics were, we had some sort of a sense of heritability. I mean, this is the reason why we resemble our parents. And even though they didn't have an understanding of heritability back then, Aristotle knew that they're positive that there was some sort of essence that came from both parents that came together in the offspring. And with Mendel's discovery of the P genetics in the 1850s and 60s, it kind of started coming a little bit more together. That this heritability were these discrete units of things, even though we didn't know what DNA was. So once this all started coming together, we had an idea of what the mechanism was of why we resemble our parents to some extent. And we know this helps determine a lot of characteristics. We know things like height and eye color are partially determined by genetics. It also tells us about more sinister things, like our susceptibility to diseases like cancer, ALS, MS. It also tells us about things that we didn't know could be genetic, like our propensity for creativity, our ability to respond to stress. So we clearly know that there's some sort of a genetic component to all sorts of these things. But what happens when we travel into the Twilight Zone? We see a whole bunch of other crazier things. We got the Cronenbergs mashing together a bunch of species DNA to form these monsters. We get humans being irradiated and growing all of these sorts of weird structures. We get dinosaurs, we get clones and super soldiers, we get humans spliced with other animals, we get weird Nazi eugenics programs. It's bizarre. And when you compare our current understanding of genetics with that seen in science fiction, it gives you this very powerful vibe. And it makes you ask the question, is our biology just a giant genetic switchboard? And hopefully by the end of this talk, you will see that more or less, yes, there is a lot of what defines us that is determined by DNA. But the catch is, we just don't know much about this switchboard right now. So in these movies today, I picked these out because I think they represent two distinct approaches to talking about genetic engineering. Before we get to them, though, I want to do a little bit of 101 into it, though. So what is genetic engineering and what tools do we use to achieve it? Next, we'll look at our first movie, Jurassic Park, and ask the questions, could we bring back the dinosaurs? And could we turn animals into quote unquote dinosaurs? And then for the last part, we'll look at the movie Gattaca and ask the questions, could we ever engineer the perfect human being? And what are the societal implications of designer babies? So as that said, let's get started. What is genetic engineering? If you look up images of genetic engineering in Google, I think it's absolutely hysterical. So this one on the left, that just looks like this person is injecting the tomato with food coloring. That is not genetic engineering. The one on the right is someone tweezering out some DNA. And you don't need to be a geneticist to know that DNA is really, really, really small and you can't tweezer it out. The textbook definition of genetic engineering is the purposeful modification of an organism's characteristics by changing genetic material. So for us humans, that would be our genome, which is 3.2 billion molecules of DNA, henceforth known as base pairs. So that is the genetic material that we would be working with. And these materials are the blueprint to what make us what we are. In the Lego box of life, DNA is the blueprint to our Lego creation. We have DNA, which is transcribed into RNA, which is translated into amino acids, and they assemble together to form proteins. And that's what we're all made of. My eyeballs are proteins, my fingernails are proteins, everything about me is a combination of proteins. And in this genetic world, the blueprints are made of just 4 types of letters. So here we have T, A, C, and G, and those stand for thymine, adenine, cytosine, and guanine. All 3.2 billion base pairs of our genome are made of a combination of just 4 letters. And in this genetic world, the Legos that we are built of are made up of a bunch of different types of materials with distinct properties. So if you were to take a look at a protein and look at the amino acid chain that it's made of, you would see that each one of these amino acids has a distinct chemical structure. Now, you don't need to know what those are, but the important thing to know is that each one of these amino acids has distinct chemical properties from each other. So a protein is made of the sum of all of the chemical properties that the amino acids have. So we're to understand humans in terms of genetics, of DNA, which encodes for amino acids, and proteins, which are made up of distinct amino acids. We can imagine our Lego creation as blueprints, which encode for materials, and Legos, which are made of distinct materials. So in terms of genetic engineering, this blueprint can be changed in a lot of ways. So these examples on the left are what is known as point mutations, and that's where you would change one base pair into another. In this example here on the far left, we have TTC, and those will be transcribed and translated into this amino acid. If we change some of these DNA base pairs, we can end up with possibly different results. And that's not the only way that we can change DNA. We can delete chunks of it. We can duplicate chunks of it. We can flip chunks of it around. We can pop it into new places, or we can move it onto entirely different chromosomes. There are a bunch of different ways that we can edit our blueprint and get different outcomes. And I think one of the coolest ways of looking at this is actually through evolution, because it is the best example of DNA change. Because whether you are a human or a fish or a fungus or a plant or an algae or a single cell eukaryote, archaea, bacteria, we all came from a universal common ancestor billions of years ago. All of the life that you see on Earth are different variations of how that DNA could change. And not only the life that we see nowadays, if we were to look through the fossil record, through hundreds of millions of years of history, we not only see the life that currently exists, but the life that previously existed, showing us all the different avenues in which DNA can make different outcomes. So I think a really good way to capture this is with the Galapagos finches. They are the literal textbook example of evolution. You have a bunch of different finches with different shaped beaks, and each of those beaks is theoretically suited for different types of diets, fruit, seeds, insects, whatever. So researchers in 2016 actually set out to answer this question. The total finch genome is 1 billion base pairs of DNA, but how many of those do you think contribute to different beak sizes? Let's take 30 seconds and think on it. Is it A, 1 million, B, 100,000, C, 10,000, D, 1,000, or E, none of the above? That song so gets stuck in my head. All right. So the answer to that is E, none of the above. The amount of DNA base pairs which are different to contribute to different beak sizes, 11. 11 out of 1 billion base pairs are contributed to the differences in beak shape. But there's a catch to this, though. There were 32,000 total differences that you could see between one finch to another. So it brings up this weird question. If there are 32,000 total differences, why is it that only 11 of them are contributing to beak shape? And this gets to another important concept of genetic engineering. What determines what traits a particular section of DNA will contribute to? It gets us to the notion of a gene. So a gene is a section of DNA that encodes for a specific protein. So our genome is 3.2 billion base pairs of DNA, but it's not all one gene. There are little blocks of it, maybe 100,000 or 10,000 base pairs that contribute to a specific protein. So in terms of our Lego box, I think it's nice to imagine each gene like a gear in a machine. And the fact is, we know a lot about the gears now. So on the right here is an example of what's called a gene regulatory network. So each one of these boxes represents a distinct gene, and the arrows representing the relationship it has with other genes. All of this right here is thought to be responsible for the development of bones in our body. And that's probably not even the complete picture either. So we have to have all of this crazy network just to make bones. The fact is, we have a lot of gears. On our 23 pairs of chromosomes, humans are estimated to have somewhere around 12,000 total genes. That's a lot of different ways that our Lego creation is working. So what tools do we need then to change these cogs? That's the whole premise of genetic engineering. So what's nice for us, and this is kind of the framework, is that the tools basically work the same across all life. So on the left here is a picture of this like glowing jellyfish. And the reason why it can glow is because of a gene that it has called green fluorescent protein. Now, we know that DNA transcribes into RNA, translates into protein. That works the same way it does in other creatures. So we could take the DNA for the green fluorescent protein gene, stick it in a fish, and then we have a glowing fish. Like, it's awesome, and it also serves a really important purpose for research. So this green fluorescent protein gene is actually hooked up to type 2 collagen, which is involved in skeleton development. So wherever we see green glowing, we know that is where type 2 collagen is turned on at. So it's a useful marker. And we need a way in this universal system to transfer information between systems. And that's where viruses and plasmids come in. They're both examples of non-living things. And we hear a lot in the news nowadays about viruses, but not many people know about plasmids. Plasmids are these small circular rings of DNA that exist within inside bacteria, but don't comprise part of its chromosome. So it just kind of shares DNA with the bacteria. So both of these things, viruses and plasmids, are capable of transferring genetic information between organisms, and plasmids and viruses are very easy to modify. So we can add our modifications to them, stick them into a different creature, and hopefully we'll be able to modify that creature. So we have a way to transfer information, but we need machinery to do this. And imagining in human terms, you would need human proteins to do human jobs. So I frankly don't want to grind up a bunch of humans to get human proteins. But we can use bacteria to do that, though, because it's universal. So on the left here is an example of what's known as CRISPR-Cas9. It's very common nowadays because it's a very new technology. It actually takes advantage of a bacteria immune system in order to genetically modify things. We customize this section of RNA here. It pairs with what is known as the Cas9 protein, and we can genetically modify any particular site that we want to in the genome. And on the left here is an example of a species called Agrobacterium. So for a lot of methods, plants are very hard to edit. But agrobacterium is a natural species that infects plants. So we could stick changes into agrobacterium and then stick it into a plant, and it could make those changes. So I think a good way to actually show like this whole process through are looking at examples of modern day genetic engineering. So we're going to get to talk about GMOs. So the one example that I'm really going to break down into detail is how we've genetically modified plants to fight off insects. So historically, humans have never been safe when it comes to protecting our food. We have records going back 4,000 years of people bombarding their plants with sulfur and arsenic to try to get rid of bugs. It's just only in modern time that we've known that those chemicals aren't very good for you. And not only us, or the environment as a whole. When you bombard plants with chemicals, those chemicals seep into the soil, they can go into the water, and they can damage more things than just those pests that we were trying to get rid of. So using our knowledge of genetic engineering, what if we had a way to mess with the pest DNA? When I see a living creature now, I see gears, lots and lots of gears that we can play with. And as most people know, it's easier to break a machine than it is to fix it. So let's break one of those gears. So we could make some changes to it. We could use CRISPR-Cas9 to change part of the DNA, which would change part of the RNA, change part of the amino acids, and thus the protein as a whole. Or we could just turn the whole thing off. We don't mess with the DNA, but we stop transcription from happening. So no RNA, no amino acids, no protein. Or we could turn it off at the translation phase. The results end up the same. There is no protein, and that's what everything about us is. So the pest target of interest is the SNF7 gene. So SNF7 is important for sorting transmembrane proteins in cells. Beautiful figure. But what does that mean? Obama doesn't know, you don't know, and frankly, I don't really know that well either. The big translation there is less SNF7 or no SNF7 messes up the pests. On the left here is an example of these normal bugs, and on the right is what happens when they have reduced SNF7 activity. They're all shriveled up, they die. It's a good tool. So there's our solution then, right? We genetically modify each larva to have a messed up SNF7 gene. We stick a needle in it or something and do that. All the billions of them. Frankly, I don't want to be the one that goes out there in the cornfields and stabbing bugs with needles to try to get rid of them. There has to be a better solution, right? So what if we modified the plants instead? Because that's the whole purpose of this, right? We're trying to stop the bugs from eating our crops. So we modify the crops. So what we can do then, since we're not directly editing the bug, is we can build a weapon that attacks the bugs. And in this, we will use double-stranded RNAs. How they work is we customize it to a particular sequence. It will pair with it, and then with the help of proteins, it will cut up that RNA. So we could design a double-stranded RNA that would attack SNF7. So it would act at this step. We're not changing the DNA or the RNA, but we are preventing translation from happening. So here's a complete breakdown from start to finish of how this example of genetic engineering works. We design a double-stranded RNA for SNF7. We stick it to a plasmid. We stick the plasmid in agrobacterium, infect the plants with agrobacterium. So now the plants will produce SNF7 double-stranded RNA. The rootworms will eat the plant. The double-stranded RNA will do its job. It will attack the SNF7 RNA. And we've got sad, shriveled up worms. So awesome. But what about humans eating these plants? Because humans have an SNF7 gene. So like what happens if that double-stranded RNA tries to target us? What's nice though, is that this area highlighted in orange is the highest similarity between the human and rootworm SNF7, but it's only about 60% similar. So in terms of something that's supposed to have near perfect alignment, we're giving it a D- right off the bat. So that's not very good, but we can play it safe. So the solution then, let's just target somewhere else. We don't have to target the whole gene. We just target a specific part of it. And what research has shown is that humans could eat billions of times the dosage found in these plants and still have no side effects. But then you might be asking me, Zach, the human cells are okay. What about our gut bacteria? Well, bacteria don't have an SNF7 gene. So there's no risk to them when we're eating these GMO plants anyways. So huzzah, it works very nicely. And so there are some other examples too. We've engineered our crops to be resistant to herbicides. So normally we use glyphosate or Roundup when we're trying to kill weeds, but we don't want our plants to die for it. So we engineer the plants to be resistant to glyphosate. But then there is that caveat again. What about the gut bacteria? And then this one's problematic because some bacteria would actually be affected by Roundup. So it's important that despite our plants being resistant to it, we don't use too much of these herbicides because we could still end up being affected by it. We know that our gut bacteria play a way bigger role in our health than we previously thought. So even though human cells are okay, our gut bacteria cells might not be okay. And for my last example, I wanted to talk about golden rice, which is actually a genetically engineered strain of rice which is designed to produce vitamin A, something it didn't do before. And this, I think, is the most important one because it is an incredibly important tool in the developing world. Rice is a staple crop for almost 70% of the world's population, and vitamin A deficiency is a serious problem in the developing world. So golden rice has helped hundreds of millions of people across the world get their daily source of vitamin A. So overall, over the past few decades, we have learned exponential amounts about genetics and how to manipulate it in living things for all sorts of purposes. So now, with that said, let's go to our movies. The first one we're going to talk about is Jurassic Park. For those of you that haven't seen it, it's based on a company that manages to get dinosaur DNA. They built a park of dinosaurs, and it's amazing. I genuinely think this is the movie that got me into this field in the first place. I know my parents tell me stories of when I was like 3 years old and I watched this movie and my mind was just like this movie hits very home to me. But we're here to talk about the genetic engineering. So there are 3 things I want to focus on in this movie. 1, they recover dinosaur DNA from mosquitoes and amber, and they fix it with frog DNA. 2, they modify the dinosaurs to not produce lysine, which is a type of amino acid. And lastly, they modified the dinosaurs to all be female. So let's break each of these down. Number 1, can we modify animals to not produce lysine? Absolutely. We could use CRISPR-Cas9 and we could break a gene which is involved in lysine synthesis. But there's a catch to this. Vertebrates already don't make lysine. This is kind of the basis of like why we have to have certain things in our diet is because our bodies automatically don't make those amino acids. And this problem arises in Jurassic Park too. They say that they modify the dinosaurs to not have lysine, but then the dinosaurs just go and eat things. So it's kind of like, I don't know, in real life, it's not actually necessary, but whatever. It's possible, we don't need to. Can we modify animals to all be female? Possibly. So the process of sex differentiation, in which we have an undifferentiated gonad, which turns into either testes or ovary, is a very, very complicated process. On the left here is an example of part of that gene regulatory network. Each one of these abbreviations is a different gene. So when we think genes, we think gears, lots and lots of gears. And when there are lots and lots of gears, that means there are lots of things that can change in this system and not give the results that we expect. And that's what we see. Human sex differentiation isn't binary. We don't end up with, oh, if you have two X chromosomes, you have ovaries and a vagina. If you have an X and a Y chromosome, you have testes and a penis. No, that is not fixed. It isn't binary. In fact, there are 16 known variations of chromosome number in humans. And not only differences in total chromosome number, but just how those chromosomes end up assigning sex. You can have XX female, but still XX male or XX intersex, which brings me to a very important aspect of this. Intersex is the discrepancy between internal and external genitalia, regardless of chromosomes. We know there are lots of genes that are involved in this process, so we can imagine how if one of these gears gets modified, we can change the total outcome. It doesn't need to be fixed to 2 fates. There's actually an entire spectrum of different outcomes we could get. So in our dinosaurs, we would need to modify all or enough of the genes required in the sex differentiation network. If we shut off enough of these genes here, the blue genes, which would contribute to testes, theoretically, the dinosaurs would always develop ovaries, which in the movie doesn't always happen because, as Jeff Goldblum will tell us, life finds a way. They change sexes. And so not only would we be modifying dinosaurs to be female, but this work has really cool real-world applications in designing physical therapies which are more applicable for the intersex community. An important thing to remember with a lot of these genes is that they don't only have one job. For example, SOX9 is very important in the differentiation of the testes, but it's also a critical gene in skeleton development. So you can see that if someone had mutations in SOX9, it would not only affect sex differentiation, but their bone growth and cartilage growth. So understanding more about how sex differentiation happens can also give us more insights into other physical phenomenon too. So number 3, can we recover dinosaur DNA from amber? This is the one that's really sad for me because probably not. It's a very, very unlikely. DNA breaks down fast, geologically speaking. And what I mean by breaking down is breaking the bonds that hold each strand of DNA together. So if you look down at this alphabet soup of A, T, C's, and G's, you couldn't tell me what the sequence was supposed to be. And we think that the half-life of DNA is about 500 years. So 66 million years, 500 years, a very big gap between them. But you might be asking me, well, it's because of the amber. But what is the oldest DNA we've consistently recovered from amber? Consistently is the catch there. So in the movie, they claim they find DNA 80 million years old. But in real life, we can recover 6-year-old DNA. Amber doesn't do a very good job of preserving DNA, actually. It's really, really bad at it. But there are exceptions to the breakdown, though. So, for example, we found DNA in ancient human teeth that dates back to about 8,000 years. We found frozen woolly mammoth DNA, which dates back to about 30,000 years. But what are the extremes of this? What is the oldest DNA we've ever found? So let's think about this a little bit. A, 50,000 years, B, 740,000 years, C, 1.4 million years old, or D, 10.6 million years old. So we got a B and an A. So the answer is C, 1.4 million years old. We've recovered it from plankton on the sea floor, which we think is theoretically the perfect environment for preserving DNA. Very high salt content, very low temperature, very low oxygen, which theoretically should preserve it very nicely. But that's the best we've ever done. We still need to find way older DNA on the total geologic time period going hundreds of millions of years. Here is where our oldest stuff is, 1.4. To get even any dinosaur, we need to go back 66 million years ago because that's when they died out. But not all dinosaurs lived at that point. To get all of the dinosaurs that we see in Jurassic Park, we would need to go back 193 million years ago to get all of that DNA. So minimum, we need 40 times older than the best scenarios on Earth, right, that we think. So it's very unlikely. And then also another interesting question. Even if we could get dino DNA, birds are the closest related living animals to dinosaurs. So if you think back to those old school depictions of dinosaurs, just these sluggish monsters, we depict them like reptiles. But when we look more at their skeletal anatomy, particularly their feet, their pelvis, and their skull, we see that they actually have more in common with birds. So then that makes me ask the question, why not use a bird to fix it instead? So on this phylogenetic tree, here are all vertebrates. So birds are closer related to dinosaurs. And the weird thing is, dinosaurs are actually closer related to humans than they are to amphibians. So the frog logic makes no sense. And it even's worse because in the movie, that's the reason why the dinosaurs switch sex, is because they use frog DNA, and frogs can change sex. So it just seems like a double whammy. Like let's use the DNA of the animal that's closer related to it. So other than bringing dinosaurs back to life, there actually are projects on board right now for resurrecting the woolly mammoth. So at Harvard University, George Church, this guy on the right here, is actually trying to bring back woolly mammoths. So he's trying to recover the woolly mammoth DNA, put it into an elephant, and then stimulate it into an elephant cell and stimulate it to try to turn it into an embryo. It hasn't happened yet, but I have very high hopes for this project, and it would be really, really cool to bring back woolly mammoths from extinction. So overall, how do we rate the genetic engineering in Jurassic Park? So number one, they modified dinosaurs to not produce lysine. Confirmed. We can totally do that. We just don't need to. Like, it already isn't necessary. Two, modifying dinosaurs to all be female. Plausible. We just need to know a lot more about the sex differentiation network. And lastly, and probably most importantly, finding and fixing dino DNA. Busted. We're just not going to be able to do that. So we probably won't be resurrecting a dinosaur anytime soon, sadly enough. But that makes me ask the question, could we just make a dinosaur instead? If we know that birds are the closest related creatures to dinosaurs, let's modify birds. So there are a couple of things that people have actually talked about with respect to this. So number one, could we turn feathers into scales? And it's actually more feasible than you'd think, because scales and feathers, and even our own hair as well, are structures which are all known as epidermal placodes that use similar genes in their development. So particularly beta-catenin and retinoic acid. So a really cool example of seeing how close these are is on this figure here in the right. So in the top left here, this is a normal chicken foot. And on the chicken foot, it has scales. But if you treat these scales with retinoic acid, they actually start transforming into structures which more resemble feathers. So there's enough overlap between the programs that if we knew all of the circuits for it, we could change them. So that's our solution then. If we modify the epidermal placodes in chickens to have genes which are more similar to those seen in scales, we could theoretically turn bird feathers back into scales. And for the feasibility of this, it's actually possible, and we're somewhat close right now. We actually understand a lot of it, but there's still some work to be done. So number two, could we get rid of the beaks and add teeth? So if you ever look inside a chicken mouth, the beak itself has no teeth in it because it actually covers the teeth during development. But like on the right here is an example of Archaeopteryx, thought to be like one of the first like proto-birds, and it still had teeth. So how would we do that? There's actually an example in real life of this happening in chickens, and these mutants are known as Talpid-2. So they develop teeth in place of a beak. So on the left here is a picture of a normal chicken embryo, a beak, no teeth. And on the right here is an example of a Talpid-2 mutant, which has these sets of teeth here and no beak. So the reason why that can happen is because Talpid-2 helps a gene known as Sonic Hedgehog perform its job. Kid you not, its name is legitimately Sonic Hedgehog. It's super cool. But the problem is Sonic Hedgehog does a lot of jobs. So these Talpid-2 mutants actually die before they're born. So we can imagine that. If Sonic Hedgehog does a lot of jobs, we mess up Sonic Hedgehog, a bunch of things get messed up as well. And this is an important premise in genetic engineering. Sometimes we can mess up too many things. So the solution then is to modify the right components of the Sonic Hedgehog network to stop the beak from growing, but not to cause bigger problems that would kill the embryo. So these are possible, but we need to learn a lot more about it. There's just so many things that Sonic Hedgehog does. It requires a lot of nuance. And so lastly, could we give birds longer tails? So we got a little stub of a tail here in this chicken, and on this dinosaur here known as Deinonychus, it has a very big tail. So we actually know genes that are involved in this specific process. The gene HOXB13 is known to stop tail development. So on the left here, this is a normal mouse tail here. And on the right, this is a mouse tail that has decreased HOXB13 activity, and it's a longer tail. Conversely, we've noted that if you have extra HOXB13 activity, the tail is even shorter. So there's this very direct connection between HOXB13 activity and tail length. So if we turn down HOXB13 activity in the tail, it might possibly grow longer. And this is very feasible. Editing one gene is a pretty straightforward process. It does more things than just the tail, but those are things that are a lot easier to understand than something like Sonic Hedgehog. So what is the future of the Chickenosaurus? If you look up pictures online, I think they're just really goofy. It just shows you like two extremes of like someone very obviously just photoshopping chicken stuff onto a velociraptor. And then you have someone like, this seems like a very realistic thing we would make. So I think we're somewhat close to making a Chickenosaurus, but there's still a lot that we need to do. We need to understand, probably most importantly, how to get rid of beaks. Beaks are a really big pain in the butt. So yeah, next we come to our second movie, Gattaca. So for those of you that haven't seen this movie, it's based in a dystopian futuristic world in which every aspect of human existence can be like determined ahead of time, and you can genetically engineer the perfect baby. So in this movie, our main character is Vincent Freeman, and his parents decided not to genetically modify him. And so in the world, he faces a lot of persecution because of it. And so it stops him from doing what he wants in life. So the whole movie is him trying to overcome these odds in order to get his dream job. So in the movie, they don't really talk about the fine details of it like they do in Jurassic Park. So I wanted to have a couple more general discussion points that are focused around the plot. So number one, can we modify complex human traits like height and strength? Two, what are the socioeconomic contexts we need to consider with genetically modifying humans? And three, how do we reconcile genetics with identity? So for the first one, can we modify complex human traits like height and strength? So in the movie, even though Vincent wasn't genetically modified, his parents decided to genetically modify his younger brother, Anton. And in the movie, it's made really, really clear that like Anton is like taller, faster, stronger than his brother. And so that's like this really big deterministic question of like, can Vincent actually ever be better than his brother if his brother is like genetically better? But like, what do we know about those traits right now? Could we modify a trait like height? I would say definitely not. We are nowhere close to that. And so then that asks the question then of like, what is the genetic basis of height? If I can modify a chicken to have a longer tail, why can't I modify a human to be taller? So we have to think first in total genetic differences, and then we can go from there. So like we've talked about, the human genome is 3.2 billion base pairs, and humans are roughly 99.6% similar to one another genetically. So that means that between any two non-related humans, we should expect around 12.8 million DNA differences. But which one of those contribute to height? You make a study that tests that. You measure the height of a bunch of humans, you sequence each of their DNA, and then theoretically, you should be able to find DNA differences in humans which might be connected to their height. And what you get with that is a plot which is known as a genome-wide association study. It's a very complex result, but I can explain this a little briefly. So on the x-axis here, we have different chromosomes, each one of these representing a chromosome pair. And on the y-axis, it represents a log probability. So in these sorts of studies, we don't connect a certain genetic trait to that physical trait. We do the likelihood of it being coincidence. So a good example here on the third chromosome, there are all of these points here that are very high likelihood. So what that means is that it is very unlikely that these points are there by coincidence. Like it indirectly suggests that they are involved with the trait. And so thinking back to our HOXB13, that's one gene. This is a lot of points to look through. And in fact, the researchers in this study suggest that there are at least 610 genes involved in determining height. That's a lot of stuff to modify. And you can imagine there's lots of ways that that can go wrong. So, and if we break it down, it makes a lot more sense. What you're calling height is actually differences in muscle, skeleton, tendon, ligaments, veins, arteries, nerves, skin. And each one of those things requires its own genes to operate. So to change something that requires all of those things, it's a gigantic concert of different genes involved. So the fact of the matter is we are still a long way away from being able to genetically modify humans for more complex traits. And so that brings in a more ethical question. What are the socioeconomic contexts we need to consider with genetically modifying humans? So first off, an important thing to remember is that environment can skew our interpretation of genes involved with traits. So let's go back to that GWAS study again. Of all of these markers that we think are associated with height, this is a hypothetical scenario, but let's think about it. What happens if all of these points came from a couple of wealthy families? They had very easy lives. They just had plenty to eat. They just never worried about anything. What if all of these points came from people with a high protein diet? For whatever reason, they just ate lots and lots of meat and nuts and cheese and whatever. They just had lots of protein, so maybe they grow faster or something. And then what if all of these points came from people that had taken performance enhancing drugs? So those points don't actually indicate anything about their genetics. It's the performance enhancing drugs that gave them a taller height. So now all of a sudden we know a lot less genes that are associated with this trait because we determined that a bunch of our genes are actually just environmental coincidences. And so the value of traits is subjective too. Imagine if we were doing a QTL, or imagine if we were doing a GWAS study for intelligence. Would all of these people be intelligent in the same way? Is the scientist smart in the same way that the mechanic is, or smart in the same way that the musician is? You can imagine that all of those scores would be different on the test, and so you wouldn't actually be able to find a genetic basis for intelligence. And so a more scary question that we have to think about is, what problems will arise if genetic modifications can only be accessed by a certain group of citizens? If it costs $200,000 to genetically modify your baby, the majority of the population isn't going to be able to do it. And then you would have a legitimate rift in society in which the ultra wealthy are able to continuously genetically modify themselves, whereas the rest of us won't be able to. Or we go broke trying to do so. And so this brings in a really nasty side of genetics history that we have to reconcile when we talk about these things. And that's what's known as eugenics. So I think it's the culmination of when genetics meets politics. So these three people here, Francis Galton, Charles Davenport, and Ernst Woden, are considered some of the main founders of eugenics. And Francis Galton was even a relative of Charles Darwin. But when he saw the basis of inheritance in genetics, he wanted to connect it into a bigger societal picture of like, how should we use this knowledge to make humanity better, which on its surface sounds like a noble goal, but there are problems with this. Even at that time, they had an incomplete understanding of this scientific phenomenon. They had ideas about what the genetics were, but they didn't know the details of it. We still don't know the details today. And this was guided by this notion that science can exist without bias. This just is impossible because we as humans intrinsically have them. So what ends up happening then is, as natural humans, we have a worldview which is biased to our own experiences and preferences. And then it ends up using science to justify one's worldview, which can get nasty very fast when we're talking about how we're going to change society to meet those needs. So these are a couple quotes that I pulled out from prominent Americans in the early 20th century. I'm not going to spend a lot of time here. You can pause the video and look at them on your own, but they are disturbing. The one I want to focus on though here quick is by H.G. Wells. He's the writer of War of the Worlds. It is in the sterilization of failures and not in the selection of successes for breeding that the possibility of an improvement of the human stock lies. That is disturbing. But that's just one person. What happens if an entire political system thinks this logic in order to make their society better? And the first one that I can think of, you would be surprised I wasn't going straight to the Nazis, was actually in the United States and in California's forced sterilization law in 1909. The premise of it was to sterilize what they quote unquote called the mentally inept, but that definition was very stretched and very racially biased. The fact of the matter was, during its existence, it disproportionately affected people of color a lot more. And it's horrible. This all happened without these people's consent. And this was all based on a racist ideology. And we're going to its logical conclusion with what happened in Nazi Germany. So you remember this guy here, Ernst Röntgen, one of the founders of eugenics? He was actually the chief architect in Nazi Germany's racial policy. And he wrote a book in 1938, which actually advocated for the forced sterilization and euthanization of children which didn't have the genes that he thought were superior, which basically just meant if they weren't white. And this manifested in a really, really horrible way of people that thought that they were doing the right thing scientifically, but were actually doing something ethically horrible. And so that brings us to a really important question with all of this as well. How do we reconcile genetics with identity? What DNA we have manifests into what we are, and what this is what interfaces with the world and what the world interfaces with us. So how does what we are affect what we think about genetics? So where do we draw the line of fixing physical conditions? In the movie, Vincent Freeman has a heart condition. And it doesn't mean he's immediately going to die. They just think he has a quote unquote heart condition. And so it prevents him from getting the job that he actually wants. And so he has to fake an identity to get the job. So, like, what about conditions like cancer and ALS? Like, those seem pretty straightforward, right? Cancer sucks, ALS sucks, let's get rid of them. But what about conditions like dwarfness and deafness? These people do suffer, but a lot of what they suffer is societal structures versus the inherent conditions of the genetics. And so having a world that is more accommodating might actually help alleviate more of their pains than genetic modifications. And so where do we draw the line of fixing mental conditions too? In the movie, Vincent Freeman has ADHD. And so that was something else that was preventing him from getting a job, because they were just like thought he wouldn't be able to do the job. And so that strikes a personal note with me because I have ADHD. Like, it's hard for me to focus. I get easily distracted on things, but it helps form a part of my identity. Like, I'm creative, I'm spontaneous. And a lot of the problems that I face on a regular day with respect to it are societal structures of how I'm supposed to live. So if you were to ask me whether I would cure ADHD genetically, I would say no. And we can bring this to other conditions too, like autism and Down syndrome, that there are some negative effects with it, but the majority of the negative effects that these communities face are from societal ostracization versus something inherent with the genetics. And then there is a dark side to this too. I also have depression, and there's nothing fun about it. There is nothing that shapes my identity, and there's nothing about it that I want other people to have. If I could cure depression genetically in a heartbeat, I would absolutely do it. So it's just incredibly important that we distinguish between inherently damaging conditions and those that are made bad by societal structures. And this isn't binary either. This is a continuum. You can imagine that these conditions are caused by lots of different changes in genes. So it's not just a fix all the genes or fix none of the genes. It's something that we can look at with a bit more nuance of how these conditions actually help manifest into positive and negative aspects of identity. So what is the future of designer babies? I would say we're still incredibly far away from making designer babies like those seen in Gattaca. So overall, how would I rate the genetic engineering in Gattaca? Super unrealistic right now. There's just no way we could ever do that. We are at least decades away from doing that. So yeah, the final take-homes for tonight are that we are not yet at the level of genetic engineering seen in science fiction movies like Jurassic Park or Gattaca, but we're not as far away from it either as you would think. We are rapidly approaching that mark. And so for the Jurassic Park side, we'll most likely never bring back the dinosaurs, but we could modify living birds to have dinosaur features. A Chickenosaurus is definitely possible. And so as a society, we need to start thinking about what we're going to use these tools for when they arrive, especially when it comes to modifying humans and the ethical and societal implications that come with that. And with that, I'm really happy that you guys were able to come today. Thank you for listening. And with that, I would be happy to take questions.
All right, awesome. Thank you, Zach, so much for a great talk. We do already have 2 questions here in the chat. So again, if anyone else has any questions, put them in the YouTube chat below, and we can take a few minutes to answer them. The first question that came up was from Hunter, and they ask, what would happen to humans if our SNF7 gene were disabled?
I would say it would end up with pretty deleterious effects like it does in the rootworm. So, in terms of imagining genetics with all of the different processes that happen in our body, SNF7 does a really, really important job in like making every cell work, whether it's a brain cell or a muscle cell or a stomach cell. So, if SNF7 were messed up in humans, we would have a very bad time. So I'm gonna go to this question from Robert. I like this idea. If this SNF7 gene basically amount to the condemnation of this species to extinction, because that is a very interesting question and something that we're like anthropogenically drawing a line in the sand for. If we are modifying these plants to kill the pests, then we are saying that the pests like don't deserve the right to live. Um, I think that's a very interesting question, and I think that's something that basically I don't have the answers to. I think that is something that conservationists need to like that we all need to have a conversation about as a whole in society. um, I hope that's one of the bigger things that everyone's able to get out of this tonight, is that this topic of genetic engineering shouldn't be something that's just left to scientists. This is something that everyone needs to talk about. If I think we should mess up these pests in their SNF7 gene, but the rest of the society says no, then we're not going to do it. So like, it doesn't matter what I think at that point. It's what all of us think together.
All right, we also have a question from John who asks, how is it that one gene can affect multiple traits? Is it that certain segments affect different things, or is it the combination of separate genes that determine how the trait manifests?
That is a very, very good question. So how is it that one gene can affect multiple things? So taken outside of its like functional context, you can imagine that all of these proteins are actually doing legitimate jobs. So when we talk about, let's say, like SNF7 for example, it is involved in the process of like moving things into and outside of the cell. So in that specific context, like we're not even talking like big picture function right now, moving things between the cell, you can imagine how that encompasses multiple jobs. So another good example of that would be like SOX9. So SOX9 helps with testes differentiation, but also skeleton differentiation. Now, like testes and skeleton are obviously very different things, but you can have still overlapping features of them that SOX9 is able to do. So how SOX9 actually works is SOX9 is a protein that actually sticks to certain sections of DNA and then like turns on and off other genes. So maybe just by coincidence, the genes that SOX9 is turning on in both of these things is like an overlapping feature of both of them. Like you might think testes and skeleton are really different, but maybe they share more features than we actually think.
All right, so if anyone has any other questions, definitely leave them in the chat. We have a few more minutes, but I have a fun question as well. So, assuming we could recover DNA much older than we currently can, what species or creature would you most want to try and resurrect?
Oh, that is so not fair. That is so not fair. You know, I'd bring back every single dinosaur back. But if I had to pick just one, if I had to pick just one, I actually wouldn't go with a dinosaur. So what I would go with was this fish that existed like 300 million years ago called Dunkleosteus. It was this gigantic fish, like the size of a shark, that she was just covered in armor plates and had these like giant blades for teeth. Like it is like one of the most terrifying looking fishes you could ever imagine. And it just looks so cool. That's hands down my favorite fish that has ever existed is Dunkleosteus.
Awesome. Okay, we have two more questions that have popped up in the chat. Lucio asks, is CRISPR the only or main tool for genetic engineering?
Very good question. So no, it is not the only tool used for genetic engineering. There are lots of different ways that we can do it. The main reason I wanted to talk about CRISPR-Cas9 right now is because it's actually relatively cheap compared to other methods of genetic engineering. So CRISPR-Cas9 has its inherent problems. One thing that's really problematic with CRISPR is, if you remember when I was describing it, I said we customized a section of RNA. It pairs with a protein, it goes and sticks to stuff. So that RNA has a tendency to sometimes go stick to random things. So CRISPR-Cas9 is by no means a perfect system, but proportional to other things that we have right now, it's very cheap. So I'd say that's like one of the reasons why this tool is kind of entering the genetic engineering spotlight.
Okay, and then we have another fun question from Robert, who asks, what ethical concerns are involved with the potential resurrection of species that went extinct a while ago and seem to lack an environment to properly slot into the 21st century? And Gilly adds, yeah, where would you put them? Where would you put a mammoth or a dinosaur or a fish?
So that's a really good question, because that like ties into the ecological concept of the niche. So if you don't know what the niche is, it's like the theoretical environmental conditions in which certain organisms are like suited for. So I think with extinction, it brings up this really interesting case of like an extinction that's very soon going to happen, or maybe actually already has happened, is the black rhinoceros. So the black rhinoceros is like either technically extinct right now or basically about to be extinct in the next like decade or so. So in that case, that's like a very easy answer for your question of like, well, that niche existed very recently. But like, how far back can you push the concept of like the niche? Like, because you can imagine that like a mammoth, like, I don't know how a mammoth would do nowadays. Like, would it destroy a lot of the Arctic ecosystem because it's just these like colossal monsters going around eating everything? Like, that's it. That's an interesting question. Like, how do they fit into the bigger environmental context? Because then you could make the case of like, well, yeah, maybe I bring the mammoth back, maybe I accidentally cause 3 species to go extinct. And then you have this weird mental game of like trying to balance the extinction ratio. Yeah, that's a very interesting question. I think it's something that we would have to think about a lot more as like a civilization about what it means for certain animals to exist.
Awesome. All right. So it looks like we have answered all the questions. So thank you again, Zach, for a great presentation. And thanks, John, for navigating and helping us with all the technical things on YouTube. If anyone watching is a CU student, you can learn more about us on CU's Buff Connect site or shoot us an email at scienceunderthedome@colorado.edu. And for everyone, keep an eye out on this YouTube channel and on Fisk Planetarium social media platforms for our Spring Talk series, which will kick off in February. Thanks so much for coming. Have a great night.